Moving on to our next speaker, Taiwo, from Nigeria, from the Helix Biogen Institute, who will be talking on exploring nuclear viral capsid and early antigen proteins, an immunoinformatic approach to designing an Epstein Barr Virus Type 1 and Type 2 vaccine. Taiwo.
Hi, good morning, good afternoon, good evening, everyone, wherever we are. My name is Taiwo O, your research assistant at Helix Biogen Institute. I would like to share my screen now. So I will be discussing on exploring nuclear viral capsid and early antigen proteins, an immunoinformatic approach to designing an Epstein Barr virus type 1 and type 2. Now, to the background study, Epstein Barr virus is a virus that houses many forms of diseases, like infectious mononucleosis, Hodgkin lymphoma, and bucket viral lymphoma. This Epstein Barr virus spreads through different means. But one of the most resortable means is the grammatically innocent means, which is the kiss. So sometimes this virus can be referred to as the kissing virus, and it's a DNA virus that is of about 172 kilo base pairs. Now, different strategies have been made to create a therapeutic Epstein Barr virus vaccine, and all of these approaches have harnessed, or let me say, incorporated the envelope glycoproteins of the virus. But in 2022, there was a request for a more therapeutic vaccine due to some recent researches about Epstein Barr virus. So now, what we did here is to create a more novel and high promising therapeutic approach to design a viral vaccine that harness the nuclear protein, the early antigen proteins, and the viral capsid protein. Now, one of the research we did is that Epstein Barr virus is associated with multiple sclerosis, and one of the antigens presented to be a culprit of multiple sclerosis is the Epstein Barr virus nuclear antigen 1. This antigen has a sequence of epitope that resembles a residue that covers the myelin sheath of the neurons. And so once an individual has been contracted with this virus, the antibody produced against EBNE1, which is the Epstein Barr virus nuclear antigen 1, is also attacking the protein that covers the myelin sheath of the human neurons. So to this end, we excluded EBNE1, which is the culprit of of multiple sclerosis. Now, the research justification: all available vaccines against Epstein Barr virus only target the envelope protein and the glycoprotein too. So we harnessed the early antigen proteins, the viral capsid protein, and the nuclear proteins in this research, which means not all available vaccines have incorporated all of these antigens in the design of an Epstein Barr virus vaccine. And as I've said, about last year, a research was released, and it's centered on multiple sclerosis being caused by Epstein Barr virus. And the question is that, is the virus we all have, or we all are expected to contract, causing multiple sclerosis? To this end, we harnessed two antigens epitope, the EBNE2 and EBNE-LP, two nuclear antigens that work in transcription activators in tandem to control the expression of viral and cellular genes and really harness the construct. What we did here, the brain behind this is that these two antigens have been justified that they are two nuclear antigens that work in tandem to control viral expression. So once these antigens have been inhibited by antibodies produced by the host system, the expression of other antigens will not be able to be furthered. So we incorporated epitopes from EBNE2 and EBNE-LP in the construct of the vaccine. Now, this is our methodology. As I've said, we made use of in silico approach. So the first thing we did is to retrieve the protein antigens that we used in the construction of the vaccine. So we retrieved 10 different antigens from the viral capsid protein, the early antigen, and also the nuclear protein. So after retrieving the proteins, we predicted possible immunogenic B cell epitope and T cell epitope. Now, these T cell epitope, they are in two forms: the cytotolic T lymphocyte and the hepatocyte lymphocyte. So we retrieved the epitopes and also performed some preliminary tests on them, such as the antigenicity evaluation, the allergenicity evaluation, their toxicity, human homology, and conservancy tests. Preferentially to the hepatocyte lymphocyte epitope, we also assess the epitopes on their inducing capability on interferon gamma, interleukin 10, and interleukin 4. Now, all of these epitopes, after rigorous assessment, were further used in the construction of the vaccine. And the vaccine, which is our primary sequence, was also tested for its physicochemical property. Now, in these physicochemical properties, we tested for parameters like the total number of the amino acid residue present in the vaccine construct, the aliphatic index, the GRAVI, and also the instability index of the vaccine. And also, since we understood that the secondary structure of the protein is also important to its function, we also get the secondary structure prediction to really validate the efficacy of the vaccine. And also, we develop a tertiary construct from the vaccine to perform some other analysis, such as the molecular docking and immune simulation of the vaccine construct. Now, one of the purpose of a vaccine is that it should be able to cover a wide range of population. So to this end, we conducted a population coverage on our vaccine. So the population coverage is to assess whether the vaccine will not only be limited to a particular region of the world, but also to all regions of the world. And also molecular docking was performed, as well as the immune simulation, molecular dynamics, construct functionality tests, and lastly, the codon optimization and in silico cloning. Now, briefly to most of the results we were able to get from the research, we're able to get that 10 antigens were responsible for the progression of the virus, which is the EBNE2, EBNE3A, EBNE3B, EBNE3C, EBNE-LP, EA, BCA, LMP1, LMP2, and LMP2B. And we made use of two genotypes of the Epstein Barr virus, that is the type 1 and the type 2. And this type 1 and type 2 include strains from Akata, GD1, Mutu, AG876, and B958. Now, out of all of the epitopes predicted for the B cell, we were able to analyze 6 promiscuous epitopes. Now, the reason why we limited this epitope to 6 is that we are trying to reduce the number of amino acid residue present in the vaccine construct, as it has been established that if an infectious vaccine will only be good if it contains between 400 to 550 amino acid residue. So we only made use of 6 promiscuous B cell epitopes in the construct. Now, why are they promiscuous? Don't forget that we are working with 2 genotypes of Epstein Barr virus. So we looked for epitopes that cut across these 2 genotypes and incorporated them into the design. And one of the parameters that will make a vaccine to be effective is their range of antigenicity, how antigenic they are, because it depends on how antigenic they are that the body will be able to respond to them as a foreign pathogen. So we made use of the antigenicity score ranging between 0.5 and 1.5. Now, one other parameter that a good vaccine should have is that the vaccine should present no allergic reaction, no toxic reaction, and also the vaccine must not be homologous to any human protein. So we made sure that all of the epitopes used were no allergic presented no allergic reaction, no toxic nature, or homolog to any human protein. Now, to the T cell epitopes, as I've said in the methodology, we harness epitopes from T cell, from the CTL and HCL. So we used 4 epitopes from the CTL and 9 epitopes from the HCL. And just same way with the B cell, we made sure that all of these epitopes are antigenic in nature, and the antigenicity score ranges between 0.5 to 2.3. And they also presented no allergic reaction. They are not toxic to the human body, and also they have no homolog protein to any human body. Now, after the validation of all of these epitopes, we fused these epitopes together to construct a novel vaccine candidate, making use of some linkers. Now, one important thing about vaccine is that a vaccine must contain an adjuvant. What's the purpose of an adjuvant? An adjuvant is to stimulate the way our body recognizes a foreign antigen. For example, a vaccine now, the vaccine will serve as a foreign antigen. So an adjuvant was incorporated into the vaccine construct, and the adjuvant used was beta-defensin 3. Although this beta-defensin 3 is also expressed by humans, so we made use of the human beta-defensin 3. And the reason why we preferentially use this adjuvant is because it has been established that one of the receptor, the TLR receptor that Epstein Barr fuses or activates, beta-defensin 3 also activates that particular receptor. So the beta-defensin 3 was used because of TLR2 and TLR9, the human toll-like receptor 2 and 9, that has been established that human Epstein Barr virus activates. Now, to the physicochemical properties of the vaccine, the theoretical PI of the vaccine showed 9.91, which means that the vaccine will be slightly basic in nature. And instability index is 40.48, although this is slightly above the threshold because the instability index of any protein should be less than or equals to 40. But with these results, we can still see that the vaccine will be slightly unstable due to the index given. And the aliphatic index, 59.55. Now, the purpose of the aliphatic index is that it will tell us whether the protein, which is the vaccine now, will be able to withstand high temperature region. Now, for example, if the vaccine is later synthesized and taken to high temperature region countries, some might not be able to withstand high temperature region, but with this result, the higher the aliphatic index, the better the vaccine will be able to withstand high temperature region. So the aliphatic index of the constructs was predicted to be 59.5. Why the Gravy value, the grand average hydropaticity, is that this result will tell us whether the protein will be able to interact with water environment. So since we have a negative result for the Gravy value, the protein will be able to interact with a water environment. All Gravy values that will be able to interact with water environment must present a negative result. And the average length of the vaccine construct is 421. As I've said, a vaccine construct must be between 400 to 550 amino acid residue. Now, molecular docking of the vaccine. There will be a receptor that the vaccine will interact with to showcase an immune response or to instigate an immune response. And these are the human toll-like receptor. Now, the human toll-like receptor 2 and 9 has been established that they are the major receptor that the Epstein Barr virus activates to induce an immune response. So to this end, we also incorporated a molecular docking with human toll-like receptor 1 and 4, just to establish the fact that Epstein Barr virus activates human toll-like receptor 2 and 9. And the molecular docking result justified this result that really Epstein Barr virus can activate TLR2 and 9 by their binding affinity. We have the TLR1 to showcase minus 29.81 kilocalorie per mole, TLR2 to give minus 40.13, TLR4 to give minus 28.93, and TLR9 to give minus 35.89. So since the human toll-like receptor 2 and 9 presented higher binding affinity, that result is justified that human Epstein Barr virus can activate human tolerant receptor 2 and 9. And the result of the complex is shown in figure 1. I incorporated the complex between the TLR2 and the vaccine construct. While in figure 2, that is the 3D structure of the vaccine that is being constructed. Now, the codon adaptation index, high value, gave 1.0. That's the maximum a vaccine or a protein should give, 1.0. If it will present an underscore, it should be lesser than that. While the GC content is 60.25. The GC content of a protein should be between 30 and 70%. This will show how easily expressible they will be in an expression vector. And since we have the 60.25% GC content, the result is also validated that the vaccine will be able to be expressed in an expression vector. Now, immune simulation. The bit of introducing immune simulation is that you want to test whether there will be an immune response at all to this antigen, because we made use of the viral antigen in the making of the vaccine. So immune simulation graphs, the first one is showing an antigen and immunoglobulin count. And as the graph is being showed here, it shows that the human body simulated can respond to the antigen to produce immunoglobulin M and immunoglobulin G, which are the major antibodies used to combat viral diseases. And also, there's the second graph is showing the B lymphocyte total count, while the third is showing the plasma B lymphocyte count, and the last one is showing the CD4 cis-EPA lymphocyte count. All of these results show that the body will be able to respond to the viral antigen, which is the vaccine, when introduced to a host. And to synthesize out this particular viral protein vaccine, we perform in silico cloning using a circular DNA plasmid. So the red portion of the circle is showing the region where the vaccine is incorporated into the DNA plasmid. Now, before this, we try to back translate the protein, which is the vaccine, into nucleotide from using E. coli as an expression vector. So we made use of E. coli expression mechanism to back translate the protein into nucleotide before incorporating it into a DNA plasmid for in silico cloning. Now, in the current study, we developed a novel vaccine candidates using the viral capsid antigen, the early antigen, and the nuclear proteins with the goal of producing a therapeutic effective vaccine against Epstein Barr virus. Now, the antibodies that we produced as per the result obtained in the course of this research will be able to prevent the expression of EBNE2 and EBNLP, which are two antigens that work in tandem to promote viral proliferation. And once this EBNE2 ability to connect the cellular DNA binding ability has been successfully inhibited, EBNE1, which is the culprit of multiple sclerosis, will be stopped. So the major driving force of this research is to create or develop a vaccine that will be able to inhibit the production of EBNE1, which is the culprit of multiple sclerosis. Now, the vaccine construct, as also said, presented an aliphatic index of 59.55, which is encouraging that the vaccine construct will be thermostable in any temperature region. And also, we performed a biological function test on the vaccine, showcasing whether the vaccine will present another biological mechanism of action or not. But fortunately, the vaccine only presented an immune response and defensive response effect, while the world population coverage of the vaccine is 85.75. This is a promising result that nearly over 85% of the total world population will respond to this antigen. Now, as the IgM and IgG count against the antigen vaccine showed high count, depicting effective antibody production. And what are the results?
Sorry, Taiwo. Yes, we. I know you're about to finish, but because of time constraints, maybe you can summarize.
All right, yeah, I'm on the last slide. So all of the results thus far has showed a candidate vaccine that is not only effective, but it's also safe, non-allergic, and non-toxic to human. Now, I would like to appreciate the efforts of my supervisors, Dr. Kolawole Oladipo, the CEO of Helix Biology Institute, and Dr. Temitope Adedusi for their constructive supervision, and my co-project designer, Elegbeleye Olubamishi Emmanuel. Thank you for listening. I would like to take questions now.
Thank you once again, Taiwo, and very interesting approach. We have one question on the chat section that you can answer, but due to time, we must move on.